Welcome everyone. Today we are going to learn about ionic basis of action potential. That means which ions and ion channels are responsible for the generation of this electric event in a neuron. As you can see, this is a typical representation of action potential. I have also included one type of electric response that is also generated by neurons. Now let me explain the graph. The x-axis represents time in milliseconds and the y-axis represents membrane potential in millivolt. Now what is membrane potential? It is also called voltage gradient. Suppose this is the inside of an action, so the voltage difference inside and outside of a neuron is called voltage gradient or membrane potential. Now I have drawn this graph in 4 different colors. The blue color represents resting potential, that means the membrane potential in an unexcited or resting neurons. The value is approximate minus 70 millivolt, and this is the baseline. Now neurons receive electrical signals or electric current, and in response to that current, the membrane potential actually changes. It can goes up or down of this minus 70 millivolt. Also, I want to mention that neurons actually show 2 types of electric response. One is passive, another is active. Action potential is an example of active electric response, and this 2 type of event is called the passive electric response. When the membrane potential of the neuron is less than threshold potential, it shows passive electric response. Now, threshold potential is actually minus 50 millivolt. Another is that when the membrane potential of the neuron is less than resting potential, also it shows a passive electric response. Now, when the membrane potential increases above the resting potential, we call it depolarization. The red lines indicate depolarizations. And the reversal of the membrane potential is called the repolarizations, and for a brief period of time during these repolarizations, the membrane potential actually becomes less than resting potential. We call it hyperpolarizations. And in between 2 action potential, there is a gap of time that is called refractory period, that is 4 milliseconds. Now I want to mention here that I have divided all these events into 5 categories. Basically, I have chosen some membrane potential ranges. Like the first is the resting potential, that is at minus 70 millivolt. The second situation is that when the membrane potential is in between minus 70 to minus 51 millivolt, that means the membrane potential has not meet or exceed the threshold potential. That means in this membrane potential range, neurons will show passive electric response. So I want to know which ions and which ion channels are responsible for this passive electric response. And the third is from minus 50 to 0 millivolt. The fourth event is from 0 to plus 40 or plus 50 millivolt. This is the peak value of the action potential. And the final is the reversal of the membrane potential from plus 40 or plus 50 millivolt to less than minus 70 millivolt, that means in the hyperpolarization. So in all these 5 different membrane potential range, what happens to the ions and ion channels? At first, I want to mention the first event, that is the resting potential, when the membrane potential is at minus 70 millivolt. Basically, sodium potassium pump, suppose this is the sodium potassium pump, it works. Now the main functioning of this pump is to generate ion concentration gradient. As you can see, it degrades or hydrolyze ATP and export sodium and import potassium. As because sodium export occur, so outside concentration of sodium is higher than inside, and as because potassium import occur, so the inside concentration of potassium is higher than outside. Now what is the significance of the generation of this ion concentration gradient? Basically, ions move through the ion channels, and ions move from high to lower concentration. Suppose this is a potassium channel. So if this channel is open, the potassium will move from inside to the outside, not in reverse, because inside concentration is higher, see, and outside concentration is lower. That's why potassium will move from inside to the outside. Now, suppose these are sodium channels, if it is open, the sodium will actually come inside of the neuron because the outside concentration is higher than the inside. So this is the reason for the generation of ion concentration, and this ion concentration gradient or the chemical force is utilized by the ion channels. Now, in all these 5 different membrane potential range, 3 ion channels are very important. The first is the potassium leak channels, or that is actually non-gated ion channels. This falls under potassium invert rectifier, that is called KIR channels. Another is voltage gated sodium channels and voltage gated potassium channels. As I have mentioned, this is a non-gated ion channel. That means it does not affected, the channel closing and opening doesn't affected by the voltage or other external factors like ligands. So at first I want to know what is happening in the resting potentials. The sodium potassium pumps actually works and it actually generates the ion concentration gradient, so it works. And the potassium leak channels remain open. Okay, one thing I want to also mention that not only potassium leak channels but a few sodium and chlorine channels is also open during the resting potential, but I have not mentioned because most of the potassium leak channels remains open and very few sodium and chlorine channels remain open. So all potassium leak channels actually remain open. So what will happen when potassium leak channels will open during this period? Potassium ions will come outside of the neuron, and the outside of the neuron membrane will become positive charge than inside. So in that situation at minus 70 millivolt or resting potential, the outside is positively charged and the inside of the neuron is negatively charged. And voltage gated sodium channel and voltage gated potassium channels remains closed in that situations. Okay. Now in this membrane potential range from minus 70 to minus 51 millivolt, the sodium potassium pump does not work because as it generates the ion concentration gradient, it stops working. Now you can ask me one question that when at the resting potential minus 70 millivolt, as the potassium ions move outside, is there any change occurs in the ion concentration? The answer is no, because very little amount of potassium ions movement can generate the resting potential minus 70 millivolt. That's why the ion concentration gradient doesn't change. So as because there is no change occurs, no significant change occurs in the ion concentration gradient, so the sodium potassium pump stops its working. So in this all other situations, I'm not going to talk about the sodium potassium pumps because they stop working. So let's talk about the potassium leak channels. What happens in this membrane potential range? Most potassium leak channels remains open, so potassium ions comes outside. So one thing I want to mention here that I forgot to mention that during this resting potential, basically the potassium ions plays important role. As you can see, all the leak channels remain open, so potassium ions comes outside. In this voltage chain range or membrane potential range, also the potassium ions channels, leak channels remains open, so potassium comes outside. A few voltage gated sodium channels and a few voltage gated potassium channels remains open, so they are insignificant. So the major contributor of membrane potential is the potassium ions, remember this, and major contributor of ion channel is the potassium leak channels. Now the third voltage range is from minus 50 to 0 millivolt. During this membrane potential change from minus 50 to 0 millivolt, potassium leak channels becomes closed. All potassium channels becomes closed because magnesium ions block the passageway of the potassium ions, and voltage gated sodium channels gradually opens. So all voltage gated sodium channels gradually opens and it takes less than 0.1 milliseconds to open. Okay, and in that voltage range from minus 50 to 0 millivolt, voltage gated potassium channels remain closed. So in this situation, I can write that mainly the sodium ions is the main contributor and the voltage gated sodium channel is the main contributor for this voltage change or the membrane potential range. Now what happens during this 0 to plus 40 millivolt? That means at the peak, it could be plus 50 millivolt also, the peak value of the action potential. Here also you see the potassium invert rectifier or these non-gated ion channels remain closed. Voltage gated sodium channels actually becomes inactivated. So in this membrane potential range, as the membrane potential rises above the 0 millivolt to the positive millivolt, many voltage gated sodium channels becomes inactivated. This is the intrinsic property of the voltage gated sodium channels. Just between 0.5 to 1 milliseconds, most of the voltage gated sodium channels actually becomes inactivated. Very few remains open. But also I want to mention this, in between this membrane potential range, sodium ions is actually important and the voltage gated sodium channels is actually important. Also, you can see here that the voltage gated potassium channels actually starts to open in this membrane potential range from plus 20 to plus 40 millivolt. So as the membrane potential increases above the 0 millivolt and above the plus 20 millivolt, more and more potassium, voltage gated potassium channels starts to open and very few remains closed. Now the last event is from plus 40 or plus 50 millivolt, it becomes less than minus 70 millivolt. What happens here? In this case, potassium leak channels or the non-gated ion channels becomes open, so potassium comes outside of the neuron. Voltage gated sodium channels becomes closed. So at first the voltage gated sodium channels remains open during minus 50 to 0 millivolt. Then it becomes inactive from 0 to plus 40 millivolt, and during this reversal of the membrane potential it becomes closed, and when the membrane potential rises from minus 50 to 0 millivolt, then it actually opens. So it actually goes through these cycles: open, inactive, and close. Voltage gated potassium channels remain open until 0 plus 40 millivolt or plus 50 millivolt to minus 70 millivolt, but becomes closed. The membrane potential becomes less than minus 70 millivolt or hyperpolarized occur. So I want to mention here that in this membrane potential range, potassium ions play important role, and the voltage gated potassium channels and the potassium leak channels or the non-gated ion channels actually play important role.